In the second half of our laboratory discussion video, we will be discussing the stages of animal development, particularly the embryonic development in a frog. Our objectives are: first is to identify the stages of embryonic development. This includes cleavage, blastulation, and so on. Second is to identify the different structures associated with the different stages of embryonic development.
The first and second stage we were able to identify was the early cleavage and the late cleavage stage. The process called cleavage is where the zygote is transformed into a multicellular entity. This transformation goes through mitotic division, resulting in the formation of several cleavage cells. These cleavage cells can be referred to as blastomeres. The number of blastomeres determine whether the cleavage is at its early or late stage. For instance, if there are 2 blastomeres present, this signifies that it's still in the first mitotic division or 2-cell stage. For the second mitotic division, 4 blastomeres are the 4-cell stage. Lastly, the third mitotic division results in an 8-cell stage. In this exercise, we can easily identify the blastomeres and could see that they differ in size. Small blastomeres are referred to as micromeres, while larger blastomeres are referred to as macromeres. The third stage is the blastula stage. After the cleavage stage, the morula, a solid ball of cells, is formed. Blastulation is the procedure in which the morula's blastomeres secrete fluid inside. This will be then the fluid-filled cavity within the embryo that can be seen at this stage, which is called the segmentation cavity, but is also known as blastocele. This is a spherical layer formed by the cleavage of a fertilized ovum. The cells in the blastula arrange themselves into an inner cell mass and an outer layer as the blastula forms the blastocyst of the next stage of development.
After the blastula stage is the gastrula stage. This stage of embryonic development focuses on major cell movements and rearrangements. In this stage, the body plan characteristic of a certain species is formed. It is also in this stage that embryonic tissue layers are formed, which will later on produce the organs and tissues of adult animals. Additionally, during this stage, the primitive gut, referred to as gastrocele or archenterone, is laid out. Another key event in gastrulation is the foundation of the 3 germ layers. As the blastula folds itself, the ectoderm, mesoderm, and endoderm are formed. These germ layers aid in the formation or derivation of organ systems and structures. They are the key to the formation of different organs. For instance, the central nervous system, sense organs, and epidermis are derived from the ectoderm. The heart, urogenital system, dermis of the skin, vertebral column, and muscles in the mesoderm, and the lungs and intestinal tract lining in the endoderm. The last stage is the neural plate stage and neural tube stage. Neurulation is the process of developing the central nervous system. The central nervous system is one of the first organ systems to develop, and a vertebrate's CNS develops from a dorsally located ectodermal thickening known as the neuroplate. This structure invaginates to form a neural fold and a depression known as a neural groove, which develops into a hollow neural tube.
Just like with our different laboratory discussion video, we were able to identify the different stages and structure associated with the embryonic development by reading and watching the said references. That is all. Thank you so much. I hope you all learned with us.